All right, this is just a quick lesson to finish off where we were before. We talked about the coagulation cascade, and we were here. So a clot in the arteries serving the heart is called a coronary thrombosis. Coronary arteries are the arteries that serve the heart, and thrombosis means clot. And this can lead to a heart attack, the medical term for which is a myocardial infarction. Myo for muscle, cardial for the heart. If that clot is not in the coronary arteries, but is in the arteries serving the brain, it can cause a stroke as well. So the risk factors for heart attack and stroke are the same. They are risk factors that lead to atherosclerosis. Atherosclerosis is that hardening and narrowing of the arteries due to plaque buildup in the walls. So if that is a cross-section of an artery, then you get the buildup of this plaque in here, and that plaque contains cholesterol and inflammatory cells and calcium. And if that bursts and triggers the coagulation cascade, then this whole lumen can clot up and no blood can flow, and therefore there's no oxygen getting anywhere downstream, and then you get death of the tissue that's being fed, either the heart, muscle, or the brain cells. So the risk factors for heart attacks and strokes are the same. So there's some things that you can't change. For example, your age and your family history or your genetics. There are other things that are modifiable. So smoking is a risk factor for atherosclerosis. It irritates the endothelium and leads to plaque formation. Diabetes is a risk factor and also increases atherosclerosis, as are things like inactivity and overweight or obesity, and also high blood pressure, which damages the endothelium and increases the risk of atherosclerosis and therefore heart attacks and strokes. So that's how the coagulation cascade ties into our discussion about transport and the cardiovascular system. Jumping over now to the next part of the lesson on acquired immunity with these objectives, just a reminder that your immune system consists of two parts: the innate immune system and the acquired or adaptive immune system. We've seen this several times. And your innate immune system and the adaptive or acquired both make use of white blood cells, but different kinds. The innate makes use of phagocytic cells like macrophages and neutrophils, and the acquired or adaptive system uses lymphocytes. Lymphocytes, as we've seen, can be broken down into T lymphocytes or T cells and B lymphocytes or B cells. And we've seen that T lymphocytes can be further broken down into several types, and we've already looked at helper and cytotoxic T cells. Remember that the helper cells are like the generals that recruit others, and cytotoxic T cells attack infected or cancerous cells themselves, and B cells make antibodies. So just a reminder of all the things that we've already seen here. Both phagocytic cells and lymphocytes are white blood cells. WBCs is white blood cells, but they're different kinds of white blood cells. Looking at this further, T and B lymphocytes, or T and B cells, have receptors that are specific to individual pathogens. There's a T cell or a B cell that matches any pathogen that could get into your body. An antigen is anything that can bind to an antibody, which is basically like a B cell receptor or a T cell receptor, and trigger an immune response. And those can be pathogens, but they don't have to be. We've talked about how things like nickel or pollen or dust could be antigens triggering an immune response, like an allergy, but not actually being a disease-causing agent, so therefore not being a pathogen. Now, when T and B cells bind to the antigen they recognize, they multiply by mitosis to form a large clone of identical cells. And that's what differentiates the primary immune response, the first time you're exposed to a particular antigen, from the secondary immune response, which happens the next time you're exposed to it. You already have all those memory cells, and therefore the response is much faster. So that production of identical cells, that clone, the first purpose is to attack the antigen, fight off the pathogen, if it is a pathogen, deal with the situation. But then afterwards, some are left behind as memory cells so that you retain some immunity, and therefore your next response is going to be both faster and larger. So if the y-axis here is time and the x-axis is time and the y-axis is some measure of your immune response, your first response is going to be slow and relatively low, then you're not going to go back down to 0 matching T cells or 0 antibodies. You retain memory cells, and then the second time is going to be much faster, and this should actually be even faster than that and larger. Let's change gears here to talk quickly about antibiotics. The term antibiotics refers to things that kill bacteria, and they do that by attacking features that are different in prokaryotic cells from eukaryotic cells. So for example, bacterial cell walls are different from the cell walls of eukaryotes that have cell walls, like plants. And also remember, we learned that bacteria have ribosomes that are different. They're 70S versus eukaryotic are 80S. So attacking the cell wall of bacteria or attacking the ribosomes, that would be a clever way to attack a prokaryote but not attack a eukaryote, which of course we don't want to do if we're taking antibiotics. Antibiotics don't treat viral infections. There are antiviral drugs, but those are not, strictly speaking, antibiotics. They're called antivirals. And the reason antibiotics don't treat viral infections is because viruses have none of these things. And viruses have no metabolism. Viruses live by hijacking cells, or actually live is not a great term. They exist by hijacking other cells and getting the machinery of those cells, those host cells, to make more of themselves. So viruses have no metabolism themselves. They don't manage energy or matter themselves. They use the machinery of the host cells to replicate. Let's finish up here by talking about HIV. HIV stands for the Human Immunodeficiency Virus. An immunodeficiency means that your immune system is not working properly. And HIV infects one type of T cell, and actually that's the helper T cell. And you'll see sometimes if you read about these are referred to as CD4 positive. That's because of a protein that they have on their membrane. They have the CD4 protein, and so they're called CD4 positive. But these are helper T cells. And you can imagine that if your helper T cells were infected and therefore not functioning properly, because they're the generals that recruit all the other parts of the immune system, the rest of your immune system would not function well at all either. So infection with HIV, and by the way, HIV is transmitted in body fluids. So it is sexually transmitted. It's transmitted in semen. It's also transmitted in blood as well, and so can be transmitted in, for example, injection drug use if people are not using clean needles. So the infection with HIV results in decreased helper T cells and therefore decreased immune function and overall lymphocyte numbers altogether, and immunocompromise. Immunocompromise basically means the same thing as immunodeficiency. Compromise is not being used here in the sense of we come to an agreement that's partway between what you want and what I want. It means that it is compromised, like, as if you were to say national security is compromised. Immune function is lower here. So if your immune system is not working as a result of infection, you get these rare infections and cancers that you wouldn't otherwise get because your immune system is treating infections and scanning your body for new cancerous cells and getting rid of them. And so the condition that results is called AIDS, and that stands for Acquired Immunodeficiency Syndrome. A syndrome is a collection of different features that sort of travel together. And so this collection of rare infections and rare cancers is a syndrome, and that is called AIDS. So there's a quick look at the content that we hadn't finished in these lessons.